So good afternoon. Thank you for coming. I told Alain that he did not need to introduce me yet again, so that's a sign of familiarity. We're getting to be quite good friends, and so thank you all for coming this afternoon. The topic, what I'll focus on today is specifically how snare proteins are regulated to achieve what we know of the process, of how they're regulated to achieve the extremely rapid and synchronous release of neurotransmitters in the brain. What I emphasized really in a way the last couple of lectures is that snare proteins are fusion machines that fuse spontaneously. They're always on. So if you have a V snare in one membrane and a T snare in the other membrane, they will, without any additional influence, engage with each other and fuse the membrane spontaneously. So that's a physical chemical fact. Okay? So how does that relate to systems in which neurotransmitters or other regulatory molecules are stored in the cell and are then released? How can they use the same snare proteins which would automatically fuse them? So let's just think about the following. You have at every nerve ending perhaps a dozen synaptic vesicles containing neurotransmitters that are docked to the membrane. This is the ending of this happens to be the neuromuscular junction. Docked to the membrane within really almost tens of angstroms of the plasma membrane, containing neurotransmitters. And somehow, despite the fact that each of these vesicles contains 70 V snares, a molecule called VAMP or synaptobrevin, and despite the fact that the local plasma membrane is highly concentrated, the active zone, in the relevant T snare, syntaxin plus SNAP25, despite the fact that these molecules are at far higher local concentration than is ever found in an artificial liposome system in which they will automatically fuse within a second. Why is it, or how is it, that these neurotransmitters fail to be released spontaneously? If you think about it, that itself is a fairly amazing fact. And think about what the consequences would be if that happened. Okay? Every neurotransmitter in your brain would be released entirely within a second. Well, it would be an amazing second, but that would be it. And no thought or action or any coherent cognitive activity would be possible. So there's nothing in a way more fundamental than understanding what prevents this from happening. Now, so much so that you might even wonder if it involves the same mechanism. Okay? I remember many years ago flying to Switzerland to give a talk, and I got right off the plane, and it was kind of an international conference, and happily I had the first talk, so I was able to, I didn't have to worry about falling asleep during the earlier talks. And so I gave my talk and along the lines of snares do fusion, and it's spontaneous, and so on. Then I think there was another talk. I immediately fell asleep afterwards. But what woke me up was the talk after that, where the gentleman, I won't mention his name, stood up and said he happened to be working on release of neurotransmitters. Said that I want to tell you that everything that Professor Rothman told you is wrong. That woke me up very quickly. And he says, here we have the release of hormones and neurotransmitters. It requires calcium. What Rothman says is that his fusion doesn't require calcium. And furthermore, he says that his reactions are inhibited by GTP gamma S, but actually here they're stimulated by GTP gamma S, and so on. So the pharmacology and the physiology of exocytosis, of which this is an example, are entirely different than that of membrane growth and division in the cell, which is dependent on continuous fusion of snare proteins, among other things, of course. So what I'd like to focus on today, really, is this amazing phenomenon by which synaptic vesicles are capable of releasing their neurotransmitter content within 0.2 to 1 milliseconds after the entry of calcium into the presynaptic zone. And for those of you who may not be so familiar with synaptic transmission, I will remind you that the action potential, when it arrives at the end of the nerve, opens up voltage-gated calcium channels which admit calcium. The calcium acts as a second messenger to trigger, by some mechanism, the release of these vesicles very quickly. So then there are two related questions that I would like to address today. The first is, what prevents these vesicles from fusing in the first instance? That's number one. And number two, equally as puzzling, is how are these vesicles able to fuse so very, very rapidly when the fusion by snare proteins alone is slower than that? And as I'll come to, snare proteins alone, they're pretty good. They can fuse membranes within about a second, but not in 0.1 millisecond. So how is it that fusion is prevented? And then when it is required, as triggered by the entry of calcium, what enables it to occur so very much faster than fusion in isolated systems so far by snare proteins? Now, part of the answer comes from the state of the snare complex in these vesicles. And you'll notice that some vesicles are right up against the membrane, others are not, okay? Of the vesicles right up against the membrane, perhaps 5 to 10 of them constitute what is called by physiologists the readily releasable pool, okay? And what the readily releasable pool means is that these are the guys that are actually available to fire a neurotransmitter within this 1 millisecond range of time. The other ones are not yet able to. They will replace the readily releasable pool when it is consumed. So the problem then becomes a little more limited and better defined. What is the arrangement of snare proteins in the readily releasable pool? Well, obviously, we can't just look at it, so we can only infer it. And the best inference, there are a number of inferences along this line, they all agree, but to my taste, the most interesting and best evidence has been developed by Milton Charlton and his colleagues in Toronto over the last 10 years or so, in which he takes advantage of proteolytic neurotoxins as probes for the state of the snare complex. Now, we're all familiar with botulinum toxin and tetanus toxin and related paralytic neurotoxins as poisons. And we now know as extremely potent poisons. And what they do as a class, each secreted by a different type of poisonous bacteria, what they do, each of them, is to inhibit the release of neurotransmitters. The question then is, how do they do this? And Montecucco made a major advance by discovering that these are proteolytic enzymes, highly specialized proteases, and each of them is specifically directed to, as he and subsequently others showed, to one or another of these three snare proteins: the V snare, VAMP in the synaptic vesicle, and the T snares, syntaxin and SNAP25 in the plasma membrane. And each one will cut at a unique place, and further has a unique property in that it is sensitive to the conformation of the snare complex. So when the target snare polypeptide for cleavage is embedded in an assembled snare complex, when it is part of the 4-helix bundle structure, the sequence that is recognized can no longer be cleaved. But when it is unfolded and not yet zippered, it can be cleaved. So these toxins are very elegant probes for the conformation of the snare complex. And what Charlton and his colleagues found, both in constitutively active tonic as well as in phasic central synapses, is that one botulinum toxin will prevent synaptic transmission immediately, but when it's infused. In other words, it will cleave the snare protein VAMP in the readily releasable pool, whereas a second neurotoxin category will not do that. even at very high concentrations. However, if you stimulate that nerve ending and you then consume the readily releasable pool and then replace it with the next vesicle, from that point on, these toxins will work. And so from this, he concluded that the confirmation, well, I got to tell you one more fact. The toxin that worked in the readily releasable pool actually recognizes a sequence that is, let's say, located right here in the V snare, and the toxin that doesn't work in the readily releasable pool recognizes one that's more membrane distal. And I think those of you familiar with the system or who have heard my earlier lectures will recognize that the snare complex zippers from its membrane distal end termini towards the membrane to produce the force that pushes the vesicle into the membrane. And so what this evidence told us then is that in the readily releasable pool, the V snare vamp is half zippered. It's zippered halfway, the N-terminal domain is assembled. And you may remember I showed you an experiment with optical tweezers where we mechanically pulled apart this portion of the snare complex, and we could toggle it back and forth by releasing the force and applying the force. So this half-zippered snare pin, an intermediate that we identified in physical-chemical experiments, is probably the key regulatory intermediate for the release of neurotransmitters. So to say it differently, the snare complex is frozen somehow in this half-zippered state in the readily releasable pool, and it does not progress further. Now that is an interesting answer, in a way, as to why it can be how the release can subsequently be so fast, because the snare complex is already half-zippered, so it just needs to complete. So that can help us explain, if not fully explain, how the neurotransmitter can be released so quickly at that stage. But it doesn't explain what holds it at that stage. What would prevent this otherwise downhill, strongly exergonic energy-releasing reaction from happening spontaneously? So the questions then that I'd like to address today, and I have to say at the beginning, this is current research. I will offer you potential answers that we're actively, and hypotheses that we're actively exploring, many of which have been garnered just in the last year or so. But I will not be able to provide you definitive answers. So the questions are, because the half-zippered state is intrinsically unstable, how can this state be stabilized in neurons in synaptic vesicles that are primed for fusion in the readily releasable pool? And secondly, again, the question I raised earlier, because snare proteins generally take 0.1 to 1 second to complete their fusion from the half-zippered state, and this relatively slow speed is more than ample for almost every other kind of fusion process in biology. The release of insulin into the blood, the fusion of a transport vesicle with its target membrane, all of these processes occur on an even slower timescale than this. So this is perfectly fine, but since it takes 0.1 to 1 second for this process to complete, even from the half-zippered state, how can the completion occur 1,000 to 10,000 times faster still than this at a nerve ending? So in other words, it's probably quite important that the snare complex is frozen at the half-zippered state, but merely releasing it from that state on the face of it does not explain this still enormous discrepancy in speed, which is an absolutely essential speed for neural processing. You know, how fast does the brain process information? You can make any number of estimates of that, but a simple estimate would probably be 20 or 30 cycles per second, which is the speed at which we merge images in motion picture film. And if there are perhaps 5 or 10 synapses that are involved in doing that cognitive processing, maybe it's 5 or so milliseconds for every round of synaptic transmission, which is about the time that an individual event takes in physiological measurements. It would be measurably slowed if neurotransmitter release were slowed. So probably we would be even less stupid as a species than we are if this release process were a little slower. Okay, it's probably quite important. So these are the questions I'd like to address. Now, there are 2 proteins that I will focus on that are expressed together in our nerves and they co-localize together at nerve endings, and which have been shown by genetics and biochemistry to be necessary and sufficient, in addition to the snare proteins, for synaptic transmission. So to emphasize, the snare proteins, there are many of them encoded in the genome, and they function generally in cell biology for membrane fusion. And in particular, VAMP, syntaxin, and SNAP25 are three isoforms of snares that function together in the synaptic vesicle. And there are various isoforms of those in turn. But broadly speaking, this is a subset of snares. And it is this subset of snares that is privileged to interact with these regulatory proteins, complexin and synaptotagmin. And it is their coming together in one or another way which we seek to understand how it produces the phenomena that I just described. Complexin, complexin is a helical molecule. I'll have more about its structural biology in a moment. But broadly speaking, it lies on the outer surface of the snare complex as sort of a fifth helix. Okay? You remember the snare complex has 4 helices, 2 from SNAP25 and 1 from syntaxin. That's 3 that come from the T-snare, and 1 coming from the synaptic vesicle from the protein VAMP, the V-snare. Complexin is an honorary fifth helix that in one way or another helps regulate the assembly of this snare complex. That was known before the recent work. If you remove complexin completely, if you delete it from the genome, or if you acutely eliminate its function, 2 things happen. The first thing that happens is spontaneous neurotransmitter release goes way up. So in the ordinary course of events, we release very few vesicles without trying with a nervous impulse. But as Bernard Katz discovered many years ago, there is some spontaneous release that occurs, what he called miniatures, originally in the neuromuscular junction, miniature end plate potentials. And it was from that he was able to deduce the quantal nature of synaptic transmission. And of course, a proper round of transmission is many such quanta that are superimposed together. This is classical information. When you remove complexin, or if you mutate it in certain ways, in many different systems, all systems studied in fact, the rate of Katz's spontaneous neurotransmitter release rises 10 to 100-fold. So it's a big effect. So complexin is very important in clamping synaptic transmission and preventing the spontaneous release of neurotransmitter. But it is not a calcium-binding protein, and it does not, as far as we know, interact with calcium. So it provides part of the answer. But removing complexin also has another effect. It actually reduces the amount of evoked release, the amount of neurotransmitter intentionally released by an action potential, or artificially released by releasing calcium at the nerve ending by some other means. And it reduces it by about half. And it does another thing, which is that the release that does occur, the half that's left after complexin, comes out in dribbles. So that it comes out asynchronously. It's no longer synchronous. And of course, we need efficient and fast neurotransmitter release for cognition, but we also need it to come out synchronously. Because if it comes out in dribbles, the information will get spread in time, and correlation will no longer be possible between different pathways. So complexin is clearly a very important and also multifunctional molecule. The other molecule, besides the core fusion machinery, is synaptotagmin. Oh, and I should mention that complexin is anchored in the synaptic vesicle. Sometimes it has a covalently attached lipid that links it there. Other times it's a hydrophobic peptide that is curvature dependent and which binds to the synaptic vesicle rather than the plasma membrane. But in either case, it's concentrated in the synaptic vesicle. The synaptotagmin, the calcium sensor, is also concentrated in the synaptic vesicle. It is directly inserted by a membrane anchor, and this protein discovered by Jan and Sudhoff actually consists of a repeat of 2 domains, each of which is capable of binding calcium. A so-called A domain, closer to the synaptic vesicle membrane, and a B domain further from the synaptic vesicle membrane. Synaptotagmin is a multifunctional protein. it is actually a docking protein. I'll describe to you in a moment how the C2b domain actually binds to the plasma membrane to phosphoinositides, specifically PIP2, and in all likelihood, this is the initial interaction that fixes the synaptic vesicle close to the plasma membrane. In addition, and more fundamentally, synaptotagmin is the actual calcium sensor for neurotransmission. And this was discovered in classic experiments by Thomas Südhoff and his colleagues, who were able to systematically increase and decrease the binding constant for calcium at these sites in the molecule, and thereby ratchet up or down the calcium sensitivity of neurotransmitter release in the animal. And what that showed was that this information, that synaptotagmin in one or another way carries the information for calcium, that calcium is entered in one or another way to trigger neurotransmitter release. But how? If you remove synaptotagmin completely, then what you observe is no neurotransmitter release. So you can conclude from this that synaptotagmin is required for neurotransmitter release to enable it. It is calcium binding to synaptotagmin in one or another way must release the snares from the half-zippered state. And as I've mentioned, it initially docks prior to that the vesicle to the membrane. So these two proteins are clearly important in multiple and subtle ways, but important ways, and they are essential for synchronous neurotransmitter release. Are they sufficient? You can't test or prove sufficiency from genetics. You can only test necessity. You test, one can establish sufficiency through biochemical reconstitution, which is the approach we took to this. And what we found is that when we put together snare proteins, the neuronal snare proteins, in liposomal and in other systems, they will fuse spontaneously. But we found that complexin has the capability of blocking that reaction in a way that correlated with the genetics. I won't go into the detail now. And that synaptotagmin, when it was included in this mixture, relieved the inhibited state. So in other words, we could reconstitute to purified snare proteins calcium coupling and reduction in the spontaneous process of fusion by simply adding complexin and synaptotagmin. And what this suggested is that these two proteins together are sufficient for the key aspects. So now we know, or we think we know, out of the 100 or so proteins that are concentrated in the active zone, which to focus on the most as the key players to try to address the questions that I've raised. And one of the ways that we're going about doing this, and the way that's been the most successful so far, has been through physical-chemical approaches. Now, just a bit about the biology, which suggests that synaptotagmin may actually be even more fundamental in biology than complexin. It turns out yeast actually have synaptotagmins. You know, yeast do not have regulated exocytosis. They only have constitutive secretion. And yet they have synaptotagmins. They lack complexin. And in yeast, what we now know from the work of Scott Emmer and from Pietro de Camilli and their colleagues especially, is that synaptotagmins, or variants of synaptotagmin in yeast, are responsible for the adhesions that have long been known to exist between the endoplasmic reticulum in yeast and the plasma membrane, so-called ER plasma membrane contact sites. And as they form these adhesions in one or another way, I'll tell you how in a moment, they bring the two membranes close enough to each other to enable the exchange, for example, of phospholipids mediated by transfer proteins, to allow the physical opening of ion channels, for example, for calcium, but not so close as to allow fusion. In fact, they prevent fusion. So perhaps the fundamental role of synaptotagmins is to actually mediate the binding of one membrane to another and to get them within intimate proximity, but to prevent their fusion from occurring. And that's a theme that you'll see I'll come back to. Complexin, on the other hand, is not found in yeast, but is found with synaptotagmins in all neurons and also in other endocrine cells that have that secrete peptides and related molecules. And as I've mentioned, it's uniquely needed for synchronous release, and deleting complexin results in a massive increase in spontaneous release. So that's the background in general, and let me give you now a little bit more molecular anatomy of synaptotagmin to help you understand the structural biology that we've done. Synaptotagmin, as I've mentioned, has a B domain, an A domain, and a B domain. Each is capable of binding calcium. Interestingly, this is actually a rather stupid protein. Okay? I suspect that Professor Changeux would find this protein very boring, okay, because there is no conformational change that anyone has ever found. All it does is bind calcium, as far as we know. Okay? It's a really stupid protein. And yet, it's a necessary protein, because binding calcium triggers neurotransmission. It has another interesting feature, though, in that it has a polybasic region, which is uniquely found in the membrane distal B domain. More about this in a moment. And again, to repeat the fact, it is this region that binds to PIP2 in the plasma membrane. The synaptic vesicle has no PIP2. Only the plasma membrane has PIP2. No other membrane in the cell has PIP2. It's kind of a code for the plasma membrane. And that enables this vesicle to dock. Now, the vesicle, this protein, synaptotagmin, though it is a stupid protein, is not without some charm and merits, because it does actually change its location. It may not change its shape when it binds calcium, but it changes its venue, okay? And how does it do that? Well, the first thing it does in the absence of calcium, this was discovered by Ed Chapman and his colleagues at Wisconsin many years ago, is that the synaptotagmin domain, in this case the B domain, and uniquely to the B domain, has this polybasic region on the surface that binds to PIP2 in the plasma membrane. So even before calcium enters, the domain, the B domain, has anchored itself on the plasma membrane, pre-positioning it for what will come next, which in one or another way, to be determined, is the power stroke that activates neurotransmitter release. Okay? And what is that power stroke? What the power stroke is this switch. When calcium binds to these aspartic acid residues here in this loop, here it is bound, here it hasn't yet bound, there are flanking aliphatic residues that are inserted directly into the lipid bilayer. This can be measured, for example, spectroscopically by substituting for these hydrophobic or aromatic fluorescent probes, whose fluorescence drastically increases upon this insertion. That's really what Chapman did. And if you mutate the aspartic acid residues, for example, to reduce the binding by calcium, for example, the same mutations that Sudhoff showed control synaptic transmission in the animal, you correspondingly reduce or eliminate this switch. So in one or another way, this conformational switch in which this domain reorients, I hope you can see, by 90 degrees. It's essentially a 90-degree rotation. It has to rotate 90 degrees. It must necessarily rip the polybasic region off of the phosphoinositide. And in one or another way, this switch is driving the release of neurotransmitters. So putting it all together so far, somehow calcium binds to this domain, which is the most important domain. This is the B domain that's membrane distal. I'm now talking about this domain. The A domain also has the same switch. It's also important, but it is not essential. Okay, you get more synaptic transmission, but it is not absolutely essential. So we're mostly, but not only, talking about the B domain. Now, in one or another way, this 90-degree rotation and insertion must release the snare complex from the half-zippered state. And in some way or other, this structure of synaptotagmin, this arrangement, in connection somehow or other with complexin, must stabilize the half-zippered state, and then calcium somehow toggles the system from the one state to the other. Okay? That's the general picture. And then now that's our way of thinking about it, and then the question is how. So basic to that would be understanding the arrangement of synaptotagmin on the membrane. So Jing Wang, a very talented postdoctoral fellow, who we hired, actually, she is trained as a cryo-electron microscopist. We thought it would be interesting, to examine the state of assembly of synaptotagmin on the surface of a lipid bilayer that contains acidic phospholipids like PIP2. And astonishingly, we could find no published papers where this had been studied in the absence of calcium. So we thought, let's take lipid monolayers that have these lipids and simply add the cytoplasmic domain of synaptotagmin, this bit, to them in the absence of calcium and in the presence of physiological magnesium and see what happens. And we were stunned to see that synaptotagmin assembles into rings. You can see them here. These rings have a definite size range, but a range of sizes, so there's no uniform size ring. And to foreshadow a little bit, you'll see that they have a sort of, this is negative staining, an electron opaque center, and you can see what look like somehow protein subunits arranged circularly around them. To foreshadow a little bit, these will be synaptotagmins bound to each other by the B domain. B, 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 all the way around, 15 to 20 of them in a ring. And the size of the rings, which is roughly 25 to 30 nanometers in diameter, outer diameter, corresponds to 15 to 20 synaptotagmin molecules, which is an interesting number because the number of synaptotagmins contained in a synaptic vesicle, depending on the lab, is between 15 and 20. So there's just enough synaptotagmins in the synaptic vesicle, potentially, to bind in a cooperative way to the plasma membrane to form a ring of this kind at the interface with the plasma membrane. You'll notice that there's an electron opaque area here, which suggests to us that the lipid monolayer is lifted up into the center of the ring and rising even slightly above it. And you'll see that there's an explanation for that on a structural basis in just a moment. Now, these rings are formed in the absence of calcium, and I might mention that when we either leave out the acidic phospholipid or if we mutate these polybasic residues, which are necessary for neurotransmitter release, if we mutate those, these rings don't form. And so this is the first piece of genetic evidence that connects the formation of these rings, that and the requirement for PIP2, that connects these rings to physiologic neurotransmitter release. Now, even more remarkably, these rings will form in the absence of lipid membranes. In the complete absence of membranes, synaptotagmin will polymerize in and of itself to make very similar circular rings, and it will do this only if you add PIP2 in solution or a structural mimetic. So for example, again, Jing Wang has found, if you take short-chain phospholipid analog of PIP2, which does not form micelles and it behaves as a single molecule, below its critical micelle concentration, it triggers the formation of these rings, which absolutely depend on PIP2. So somehow synaptotagmin has been duped into thinking that it is on a plasma membrane and it then polymerizes, suggesting, by the way, that there may in the end be a conformational change. So I think it's not absolutely a stupid molecule. It seems to obey the general rules. But there's no doubt that this molecule has an intrinsic capability to polymerize. Now, when synaptotagmin rings are formed on lipid monolayers, or for that matter on large bilayer vesicles, not infrequently, the membrane which is sucked up into this ring continues to be pulled through, and the ring sometimes fails to close. And as a result, the ring snakes around, and out comes a tubule. And happily, these tubules frequently have crystallographic symmetry. And so here you see a negative stain tubule, you see a diffraction pattern, and fortunately there are many such tubules. We can average them and do image processing, and I won't take you through every detail of this, but we've been able to get numerous reconstructions of many individual tubules. We've been able to characterize them into different subtypes, and although the work isn't entirely finished, we don't have the kind of resolution we would like to have, it is nonetheless adequate for us, and us in this case, I should mention importantly, is my laboratory and Charles Sindelar's laboratory at Yale, who is a structural biologist who specializes in cryo-EM. We've been able to unambiguously fit and at least reasonably well orient the synaptotagmin B and A domain X-ray crystal structures into this arrangement. So this then is a top view. So this is as if you were looking down on the rings in the previous micrograph. And now you can see the B domain bound to the B domain going around the circle. This is a cross section, and although it's cartooned here, in actual fact we see the electron density of the tube. The tube membrane would be right here. This is the reconstruction of the tube. And so you can now see in a way how the plasma membrane gets sucked into the ring. And the reason for it essentially is if you look now from the top, let's go back to the top. The polybasic region is here. These are the residues that bind PIP2 that initially anchor the B domain to the membrane, okay, and that are necessary to form the ring, okay? So presumably they are bound to acidic phospholipid right here, oriented with its head group here and the tails here. So the point then is that the inner aspect of the ring, if you think of the ring as a coming out and back, think of it as a tire, like a car tire. Here's the inner aspect of where the hub would be of the tire, and the acidic phospholipids are bound here, which is to say the membrane needs to be sucked in to the interior in order for this to form. So the membrane is being puckered, actually, the plasma membrane being puckered by this ring. Even in the earliest days, George Pilati commented on the puckers of the plasma membrane that are found with Dock vesicles in regulated exocytosis. So perhaps, we don't know, but perhaps this is part of the basis for it. Where is the calcium binding site? So these residues here, shown here are the calcium ions that will bind. I want to emphasize this is in the absence of calcium. But if calcium were to bind, they would be here. Well, that's interesting because they're actually where we predict the interface should be between the B subunits that form this ring. B, 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 B. The A subunit, the A domains are pendant, and in different tubes they are arranged differently. So in other words, there's no specific arrangement of A, but in every tube the B is arranged the same. So what would you predict? This is the calcium binding site where the aliphatic loop is that I just told you needs to insert into the membrane in order to activate synaptic transmission. But how can it do that if it's 25 angstroms away and 20 angstroms away and oriented 90 degrees the wrong way, and furthermore buried in the interface between two B subunits? So a very strong prediction will be that when you add calcium, that this ring should be disrupted. And if that ring is disrupted, you can imagine that would enable this subunit to rotate and insert into the membrane. But maybe the key feature is less the insertion into the membrane than it is the disruption of the ring, and the disruption is made permanent by the insertion of the aliphatic residue into the membrane. Well, of course, that can be tested. And in fact, even at physiological concentrations of calcium, for local calcium near a synaptic vesicle, according to Nair and others, is between 0.1 and 0.5 millimolar during the process of synchronous neurotransmitter release. And you can see that the rings that had formed have largely disappeared in this range. Now, of course, we don't have kinetic experiments that can say that they will disappear within this range of time, and I'm not sure at all that they would be capable kinetically of fully dissociating, having the subunits fully diffuse away in, for example, less than a millisecond. Actually, I think that's probably physically impossible. But the initiation of the disruption of this ring by calcium could very well be very rapid. The canonical calcium binding sites are required. So, for example, if we take the Sudhoff mutations that prevent calcium binding, that prevent the insertion into the membrane, and that prevent neurotransmitter release, these also prevent the dissociation of these rings by calcium. In fact, interestingly enough, they stabilize the ring, so you get even more rings than normal. We don't yet have a good explanation for that, but it's an interesting observation. So, a very simple idea jumps out of this. Oh, one more thing. which is we've been able to model the interface at the B domain. So according to our structure, the interface between two B domains stabilizes the ring. And there'll be some residues on one side and some on the other side of the B domain. And we've systematically analyzed all of the residues that could be in the region, mutating them to alanines initially, essentially alanine scanning. And some of them have no effect, but several of them actually do, and they have very strong effects. And so here are a series of point mutations that are present on one surface of the B domain and on the opposite surface, 180 degrees apart, that completely prevent the rings from forming. Okay? So probably and they do this, by the way, without affecting the ability of the acidic region, of the polybasic region, to bind to the acidic phospholipids, and without affecting the ability of calcium on isolated synaptotagmin domains to switch into the membrane. So it's a very selective effect on oligomerization. So we have a very simple model then, but it's still not quite detailed enough, but let's say a simple idea comes out of this. What could be simpler than clamping fusion, by preventing fusion, than by putting a ring in between the vesicle and the plasma membrane? What could be simpler? I want to prevent the vesicle from fusing. Maybe I don't even have to interact with the snare proteins. All I have to do is put in a washer, okay, a spacer between the vesicle and the membrane. Now, interestingly, the height of this ring is 5 nanometers. That corresponds to the height of a B subunit. We know that the snare complex can half zipper, and it can be half zippered when the membrane is as far as almost 10 nanometers, not 5 nanometers. So to summarize, if this ring were to form at the interface between the synaptic vesicle and the plasma membrane, there would be no structural impediment to the snare complex, to prevent the snare complex from half zippering. But it could not zipper to completion because of the height of the ring. I couldn't imagine a more beautiful and simple general explanation than that. Now, don't get me wrong, it's not a complete explanation, but it certainly suggests that we may be on the right track. Furthermore, as I've told you, synaptotagmin has been shown genetically and physiologically to be required for the close apposition, in other words, the docking of the synaptic vesicle. And the B domain has been shown to be required, and the acidic region of the B domain, the polybasic region has been shown to be required. The very requirements for, and PIP2 is also shown to be required, all of the requirements for the assembly of this ring and the polymerization of synaptotagmin, even in solution. So unless physics suddenly disappears when you enter the cell, which I doubt it does, you would say that it's very likely that this ring would polymerize at the very moment of contact of the synaptic vesicle with the plasma membrane, providing an impediment from the very first moment. Okay? And then, when calcium enters, this impediment in one or another way is removed. I've suggested a structural basis for it, and fusion can proceed. So this is an explanation that is satisfying at one level, but at another level is clearly insufficient because it can explain how synaptotagmin works in some way, but it cannot explain really the extreme kinetics of fusion that happens after calcium enters. So I'll come back to that, but I want to make a point that it doesn't fully explain that, but it could potentially explain quite a bit. So what I'm going to suggest to you, did I skip something? No, I did not. Okay, good. What I'm going to suggest to you then is that there are two general roles that these proteins have that are sort of universal. What I'm going to suggest to you is that snare pins, their basic job is to force membranes together. They are rather stupid force-generating machines. They're pins, they pull membranes together, that's what they do. And everywhere that snares are used for fusion, that's what they do. And I'm going to suggest to you that rings of synaptotagmin are a principle that is very general for synaptotagmins, and their job is to keep membranes apart. Close, but apart. And so you can easily see in an evolutionary way how if you had synaptotagmins around that keep membranes apart, for example, in simple microbes like yeast, And if you also have snare pins that are needed generally for cell growth and for vesicle trafficking, as in yeast, as a general always-on mechanism for membrane fusion, if you simply combine the two into the same membrane, you would have the irresistible force meeting the immovable object. And you would immediately be able to step up to a primitive form of regulated exocytosis. So this way of thinking is a very, at least for me anyway, a very attractive way of thinking superficially, but quite generally, about regulated exocytosis. So, are ring-like oligomers a general feature of synaptotagments? Well, obviously we haven't tested every synaptotagment encoded in the genome, but synaptotagment 1, 2, 7, and DOCK2B, a soluble version, are all calcium sensors that in one or another way are critically important for neurotransmitter release. Each and every one of them forms a ring. And interestingly, in collaboration with Dick Camilli's laboratory, we've tested an extended synaptotagment. Now, extended synaptotagments are our versions of the yeast synaptotagment that forms the ER plasma membrane junctions, and these do the same in our cells. And this also, though it is not needed in fusion, in fact mediates plasma membrane ER junctions, and we would suggest, since it forms rings, that the same basic principle is utilized here, except not in exocytosis. Perhaps it's a pure case of getting membranes close, but keeping them apart, but within a molecular contact distance, so that some other molecule can be involved in ping-ponging between the two membranes to transfer information more efficiently or to transfer materials. Now, about a year ago, we became aware through this publication of a human, a syndrome, a neurological syndrome, in which several families were identified carrying mutations that in a typical Mendelian fashion explain their disease, and these mutations are in synaptotagmin. Synaptotagmin 1 is the major isoform that is used throughout the brain for synchronous neurotransmission. Synaptotagmin 2 is also found throughout the brain, but is probably more important at the neuromuscular junction, as best I've been able to determine from the literature. And the principal problem that these people have is a dominant form of a progressive motor neuropathy. And so these folks they can move their muscles, but they have spastic movements, and they are often weak. Okay, so there's definitely a compromise, and this can be shown in muscle biopsies, and has been shown, that there's a compromise in neurotransmitter release in muscle biopsies from these patients. So the question then is, how can this be explained on some molecular basis? Well, remarkably enough, the mutation that was found turns out to be exactly at the interface between the B domains that we had identified, and as I've just described to you. But rather than having been an alanine mutation, it was an asparagine mutation. And rather than completely eliminating ring formation, as you might expect for an allele that allows, in which the individual, though compromised, is still alive and does work, where muscles do work to a large degree, you have a loss of function, but not a complete loss of function. Nevertheless, this provides a very strong, I think, piece of genetic evidence that the formation of this ring may be important even for neurotransmitter release in vivo. Well, obviously, we're now testing a large number of these mutations in simplified systems, and also we're collaborating intensively with geneticists at the Institute of Neurology at University College London, where I also have a collaborative appointment to test this hypothesis further. So I've explained the general nature of the hypothesis, and so what are the key outstanding questions? The most important is, do these rings actually form in vivo? Well, we don't know. We have no direct evidence for this today, okay? We have, however, evidence that that mutations that prevent the formation of the ring prevent neurotransmission. Mutations that prevent the disassembly of the ring with calcium prevent neurotransmission. And that mutations that interfere with the assembly of the ring actually are dominant interference mutations in humans. Dominant interference is the human genetic term, autosomal dominance amounts to dominant interference. And we've actually shown that the mutations this very mutation that affects this disease, that controls this disease, is actually dominant interfering biochemically for ring formation in vitro. So we can explain the autosomal dominance as well. So while this isn't a proof that the rings exist in vivo in the interface of the vesicle as we propose, we think that between the biochemical and the genetic evidence, it's a very strong possibility. And we are testing this now using a super resolution microscopy, and we hope to be able to push the technique to be able to see the arrangement of the ring, of the proteins in the ring, at the interface between a synaptic vesicle and a plasma membrane. And I'm optimistic from the data that we have so far that that's exactly what we will find, but the data is preliminary. Okay, the next question then is how do half-zippered snare pins interact with synaptotagmin rings? Well, maybe they don't, okay? It's entirely possible that the synaptotagmin rings are able to form, and that the snares form outside them, as I cartooned a moment ago, and they simply are trying to zip their way in towards the center to fuse and motor, and they bump into a wall. And the wall is removed when the ring disassembles, when calcium disassembles it. That's absolutely a possibility. However, it's been well known for many years that synaptotagmin binds to the snare complex. And it binds to the T snare, the half-zippered snare complex, or the fully zippered snare complex with equal avidity, probably in the range of 10 micromolar binding constant. I think it's close to the number. And so there's no doubt that the snare complex binds to synaptotagmin, one molecule to one molecule. So the next question becomes, what if the snare complex did bind to synaptotagmin and the synaptotagmin was in the ring? What would it look like? So this is an exercise in model building. Now, the information that makes this possible very recently, emerged from the first crystal structure of synaptotagmin bound to the snare complex. And that's shown here from Axel Brunger in his laboratory at Stanford. And he shows this is the contact which he discovered between the C2b domain and the snare complex. As has long been suspected, it is the SNAP25 portion of the snare complex on which synaptotagmin docks. Synaptotagmin binds to a central portion of the snare complex, and it binds to to the two helices in this region of SNAP25 through a particular surface. Mutants that he has made, he and Tom Suthoff, in these complementary surfaces very significantly affect the kinetics of synaptic transmission in the mouse system. So this contact is very likely to be physiologically relevant. Okay? This is a side view. The zippering of the snare complex occurs in this direction, so these are the N-terminal domains that zipper first. The half-zippered snare complex is actually right here. And then the completion of zippering that triggers fusion would be occurring here. Okay? What happens when we simply drop the snare complex in this fashion at this binding site onto our synaptotagmin rings? What do we see? From the top, this is what we see. Now again, this is pure speculation. We don't know that this happens. And I have to say, it's not a perfect match. Because if we you'll notice that this is the B domain, this is a B domain and an A domain. So this is BA, and this is the central SNAP25 bound to the B. Here's another SNAP25 underneath the snare complex bound to the B. You'll notice that here's a B and there's no snare complex bound there. The reason for that is that if we tried to bind the snare complex at every site, they would bump into each other. So one thing we learned from this is that it's absolutely possible that the snare complex doesn't bind to this ring. Although we would think at the very high concentrations that seems kind of unlikely. But we also learned that sterically it's rather easy for every other, or as many as every other, synaptotagmin in the ring to bind a snare complex. So one way of thinking, and I'm just now going to take you down the line of speculation, is that as many, if there are about 15 to 20 of these synaptotagmins, so you can say that there are probably about half as many, maybe as many as 10 snare complexes that could be accommodated. How are these snare complexes oriented? Well, that part is unambiguous. Their N-terminal domains are facing outside the ring. Their transmembrane domains would be going up and down from the ring, in other words, are zippering towards the center. And if we look from the side, now we see the synaptic vesicle. We see the ring with its puckered plasma membrane. This is all drawn to scale. Here's the ring of synaptotagmons with the inner B domains, the outer A domains, and now you're seeing various views of the snare complex as you go across it. And a side view cut away, which would show this. Here is the B domain, the A domain, the snare complex, half zippered. The remaining part that needs to be zippered is inside the ring. And if it were zippered, it would clearly zipper up and drive the two membranes to fuse each other. And a very simple idea as to how this ring could prevent the subsequent zippering from occurring well, the simplest idea is that the snare complex may be, the ring, may be holding the snare complex at too great a distance. So in other words, to illustrate that, let's just suppose that that this is a snare complex, and it wants to zipper in to where the fusion is going to occur. If it's free to zipper in, it will, and we know that even one snare complex is thermodynamically capable of fusion. But on the other hand, if we hold it back far enough, then it may try to pull in, but it actually won't be able to get there until I release it, which is what calcium does when it releases the integrity of the ring, and now the snare complex zippers, is allowed to zipper. So that's one possibility. And a more subtle possibility, also synergistic, has to do with the orientation suggested by the structure. If the snare complex were pointing directly in, the force vector would be directly in, but now the force vector is largely radial. And since it's radial and symmetric, it's likely to cancel. And so the inner directed component of force is likely to be much reduced. And so in this way also, a synaptotagmin molecule can function as a clamp. So that's our current way of thinking. Now, there's already a problem with this way of thinking, okay? Because it turns out, although there are perhaps 20 synaptotagmins, in other words, one ring that can form from a synaptic vesicle, each synaptic vesicle contains 70 V snares. So, if you go through the math that I just told you, as many as 10, but not more, can probably be accommodated in the ring and therefore prevented from engaging in fusion. But what about the rest? What is to prevent the remaining more or less 60 snares in the synaptic vesicle from binding to the plasma membrane and then just fusing automatically, giving rise to spontaneous fusion? What's to prevent that? Big problem. Okay. So this brings us then to the final topic, which is how does complexin synchronize and synergize with synaptotagmin rings to produce explosive release? Now, what do I mean? Why do I, once again, to remind you, since it was a long time ago now, if you remove complexin, what happens is spontaneous release goes up 10 to 100-fold, okay? And only half of the normal amount of neurotransmitter is released. The readily releasable pool is down by half, okay? And it comes out in dribbles. So explosive release, 0.1 to 1 milliseconds, no longer occurs. You get release, it's not as much, and it's asynchronous, okay? So complexin obviously needs to be fit into this in some important way. How? Well, the first clue came from physical-chemical experiments we did in Frederick Poncet and his colleagues did, I should say, principally here, and which involved a collaboration with our group at Yale, and it's a collaboration that has prospered over the last number of years and continues to prosper. And in this experiment, which is using the surface forces apparatus that has been used so profitably at École Normale Supérieure in the physics department, the experiment consists, roughly speaking, because it's a complex apparatus, but I'll explain it in ways that I understand it, of the following. You have two lipid bilayers, each on a glass surface. And in this case, you have V snares on one, namely VAMP, T snares, syntaxin, SNAP25 on the other. And you can move them together with successive degrees of closeness with very fine control to better than about 1 nanometer. And you can read the separation by interferometry. And then there's also a fancy spring, excuse me, Fred, for this, but a fancy spring that's connected that allows you to read the force, how much it's either pulling you in or how much you have to pull back in order to separate it. And so as you approach the two, this is now without complexin, just the V and the T snare, as you start bringing them together to reduce the separation in nanometers from 15 to 10 towards 5, somewhere when you get towards 10 nanometers, the two surfaces are attracted to each other because the snares start assembling, the N-terminal domains start assembling, and the snare complex starts pulling the two membranes together, okay, just as advertised. Until they are close together, and if you push on them, you now have snare pins that are lying flat between the membranes, and you can't get the membranes any closer because you can only push so far on a 4-helix bundle without crushing it, and you can't crush it. Now what you do is you start pulling apart. And at a certain point, you have to exert force in order to separate the snare complexes. You're now doing what we did with the optical tweezers, except it's not on one snare complex, it's on an ensemble, really representing two membranes in the state of fusing. And now you pull it apart. And what we observe is that these membranes, the snare complex, zippers to a very, almost completely zippers, not completely because of steric reasons. And now you can separate it up to about 10 nanometers, and at that point, the two membranes are ripped apart, okay? And it takes a certain amount of energy, about 35 kT of energy, to rip them apart. So that measures the stability of the snare complex, the energy that stabilizes them and prevents them from ripping apart. I want you to notice the distance here, 10 nanometers. Now, if we add complexin and do the same experiment, it looks superficially similar, but actually it isn't. The amount of energy needed to rip them apart is only 15 kT, not 35 kT, and in fact they rip apart at a greater distance, meaning they were less zippered, and in fact they're half zippered. So what these two changes in parameters show is that complexin creates and stabilizes a novel state of the snare complex, which is half zippered. So remember, the very famous half zippered state that is there at the readily releasable pool, it is created artificially simply by adding complexin. So complexin, you can easily see, can reduce the release of neurotransmitter, reduce spontaneous release by stabilizing the half zippered state. Next question, how does it do that structurally? Well, what we did was we crystallized complexin together with half zippered snare complexes. How can you do that when the half zippered snare complex is by definition an unstable intermediate? Well, we artificially stabilized it by making a half zippered snare complex with half of a vamp molecule, representing only the N-terminal domain of the assembled snare complex that assembles first and which is already zippered in the readily releasable pool. So here is the snare complex with the 1, 2, 3, 4 helical bundles. This is SNAP 25. This molecule here, where are we here? Is VAMP. Yes, this molecule here is VAMP. And we actually are comparing two crystal structures. One in which a VAMP peptide is present only up to here. That's the crystal structure that Karin Reinish's laboratory solved. And it represents the half-zippered state. And another in which the full VAMP sequence is present, representing the fully zippered state. The fully zippered snare complex, in conjunction with complexin, was solved by Sudhoff and Rizzo quite some years ago, about 2002. More recently, we solved the half-zippered state. And what you observe is that there's quite a difference. Here's the complexin helix, and there are two regions of it, a central helix and an accessory helix. Now, one thing that I didn't tell you about is that I told you if you completely remove complexin, the rate of minis goes dramatically up. You can get the same effect by mutating the accessory helix or completely removing it. So the accessory helix is somehow the most important part in respect to clamping synaptic transmission. The central helix is what interacts directly and most strongly with nanomolar binding to the 4-helix bundle. The central helix positions complexin in a way that somehow allows the accessory helix to function in clamping. So that much was known. the difference between these two structures is very revealing. In the post-fusion state, when the snare complex is fully zippered, the complexin accessory helix is parallel to the 4-helix bundle. But in the half-zippered state, representing, we think, the readily releasable pool, now the accessory helix is pointing out at 45 degrees, and the central helix is interacting in the same region, but has a somewhat different set of contacts. The first thing this suggests is that the central helix binds differently to the half-zippered state than it does to the fully zippered state. In other words, the central helix by itself should stabilize the half-zippered state. Okay? That suggests that the central helix, even in the absence of the accessory helix, can help stabilize the snare complex in the half-zippered state. It also creates a different geometry, the significance of which is revealed by the crystal packing. Because here, if you look at a single half-zippered snare complex, it looks as though the accessory helix is simply shooting out into outer space. But actually, in the crystal, it is not. It is actually binding to a second snare complex. It is actually capturing the snare complex into what we call a zigzag array, which is the basic structural motif in the crystal. And it is the accessory helix that does this, and it does this by binding to the portion of the T-snare, the C-terminal membrane proximal half, where the final zippering will occur. When this part of the T-snare normally zippers with this part of the V-snare, which is not here in this case, when that happens, fusion occurs. But according to the structure, it can't happen because the accessory helix is sitting there, blocking it sterically from happening. So the open arrangement of the snare complex suggested by the crystal structure would naturally seem to suggest a trans-binding mechanism in which a complexin molecule anchored on one snare pin can reach over on the same membrane and bind to another snare pin to sterically prevent the completion of zippering by its V-snare and therefore clamping membrane fusion. And quite possibly this is what was forming in the surface force apparatus at the interface between the two membranes. And of course, this can happen over and over again, and in its lowest energy state perhaps could be arranged in a zigzag array such as what we see in the crystal. Well, the first thing, you have to worry about whether this is an artifact of crystal packing. To test whether this is so, we designed several mutations. For example, we have wild type means the normal, this is the central helix and the accessory helix of complexin. This is the physiologic wild type sequence. We made mutations that should disrupt that sequence, and we made other mutations that should actually increase the binding of, or superclamp, that should increase the binding of this accessory helix to the T-snare, based on the structure. Together with Fred and his colleagues, we went back to the surface force apparatus, and indeed, we found that the non-clamping sequence does not form this state. The wild-type sequence, as shown, does, and the superclamp actually forms exactly the same state, but at 5 to 10 times lower concentration of complexin. So we've made the right mutations, and they have the effect in the isolated system of preventing or stabilizing, or even more stabilizing than normal, the state. What happens when we measure binding in trans in vitro? Now, I don't have the time to explain this assay in detail, which is done by isothermal calorimetry, but suffice it to say that when you mix the complexin and you design a system in which it can only bind in trans to the T-snare, to the terminal portion of the T-snare, to which it is bound in the crystal, when we measure the binding constant for that interaction, first of all, we see it, and in solution, with the wild-type sequence, it occurs at about 15 micromolar binding constant, which is, it seems pretty high, but the local concentration at the vesicle membrane junction is estimated to be closer to 1 millimolar. So this is likely to easily be physiologically relevant. If we mutate away the binding capability, it prevents the half-zippered state in the SFA experiment, and it also prevents the unimolecular binding in solution. And furthermore, when we use the mutation that non-physiologically stabilizes the half-zippered state, it actually binds with a higher affinity, 2 micromolar as compared to 16 micromolar in the unimolecular transbinding system. So there's no doubt that the complexin accessory helix binds directly to a T-snare even in dilute solution. So this is in no way as an artifact of crystallization. Furthermore, in collaboration with Troy Littleton, we've tested what the effects of some of these mutations might be on neurotransmitter release in flies at the neuromuscular junction. So essentially, what Troy and Richard Cho did at MIT was to redo the Bernard Katz experiments, the classic Katz experiments at the neuromuscular junction, but rather than Katz working on cats, which is what Katz did, Katz experiment was done on flies, okay? So we used cats to catch flies, as it were. Now, this is the frequency of miniature end plate potentials, spontaneous releases per second. It's a few per second in an ordinary neuromuscular junction. If you knock out the complexin gene as advertised, it goes from perhaps 3 or 4 per second to perhaps 70 or 80 per second, an enormous increase. If you now introduce the mouse complexin, normal gene, it now substitutes very respectably for the normal, but not perfectly, for the physiologic Drosophila, and drastically lowers the frequency of spontaneous release. If you break the continuity of the helix in complexin, if you prevent the accessory helix from folding to a helix, it no longer clamps spontaneous transmission. If you remove the accessory helix but leave the central helix, you still get proper neurotransmitter release, normal evoked release, but now you get about half as much, I should say, neurotransmitter release, but most importantly, there's no clamping. And if you use the super clamp mutation, that with much higher affinity stabilizes the trans clamping interaction in vitro that I just showed you, it actually lowers clamps even better than the wild type accessory helix sequence in this in vivo situation. So this would, on the face of it, seem to provide very strong evidence that the interaction between the accessory helix in trans from one snare complex to another is vital for producing, for clamping spontaneous neurotransmitter release, which at the very least lowers the noise at the synapse to allow the synaptotagmin-dependent signal. But as the name complexin suggests, it's actually more complex than that. And so I'll give you a couple examples of the complexity of complexin. Because if you had 5 of my colleagues who work on 5 different systems in here talking about this, they would each emphasize one or another thing. There are points in common among that I've emphasized about the biology of complexin and the biochemistry of complexin. To my knowledge, everything that I've just described is general. But there is a lot of complexity and seeming contradictions from one system to another. And so there is no consensus as a result from genetics and physiology around complexin. And that's why my hope has been that structural biology could forge a deeper level of understanding, and then an understanding of the genetics might result in the same way that the confusion that you remember the talk where I fell asleep, and it seemed that neurotransmission and vesicle traffic generally had to be different. Well, maybe they're not really as different as they seem. There's a diversity of regulations superimposed on something more fundamental. Well, here are some examples of this. The accessory helix. As Dittman recently showed, you actually can put any helical sequence you want in there for the accessory helix. You have to have an accessory helix. This is in worms. You have to have one. It's got to be the right length. It's got to form a helix, but it can be positively charged, negatively charged, or no charge. Doesn't matter. And a scrambled sequence works perfectly well as long as it forms a helix. So it's a steric mechanism. Well, that's broadly consistent with what I've told you, a steric mechanism that prevents zippering, but it means you don't necessarily need a high affinity for that interaction to work, for the accessory helix to clamp. Now here's the other one that's actually a little more difficult, and there are many examples of this sort. Deleting the accessory helix. Well, I told you that deleting the accessory helix massively enables spontaneous release. But actually, deleting the accessory helix, at least in Drosophila, has no effect on evoked release. None. Okay? Now, it may slow it a little bit. The time resolution of these studies is not huge. But the efficiency of calcium-dependent evoked release is 100%, and superficially the timing is very similar. Okay? How can you explain that? Well, on the face of it, the genetics suggests that complexin functions, at least the accessory helix of complexin, is important only in preventing spontaneous release, and perhaps not as part of the greater pathway of the calcium switch that enables the snare complex to complete. But on the other hand, if you take away the central helix of complexin, okay, or all of complexin, then what you lose, then you actually end up with dribbles of vesicles coming out with half the efficiency, as I've told you before. Okay? So complexin in the accessory helix seems to work. It may be the accessory helix and the central helix, though in the same molecule, function in two ways, which is to say, it may be that complexin molecules function in two supramolecular complexes, one to clamp spontaneous release, and the other to somehow facilitate evoked release. Okay? So it is a little more complicated, and just in summary, to let you know how we're thinking, because we have, it is on the face of it quite complicated. So I just want to point out something. I had lunch with the students today who were telling me about some of their troubles. Troubles are good for you, okay? The famous biochemist Ephraim Racker used to say, he had something he called the TAGFY, tag fee philosophy. Troubles are good for you. And I have found over and over again in my experience that when you are getting into trouble, when everything seems good and suddenly, the rug gets pulled out from under you and you thought you understood something. I was here in November and I showed a model and I said, I think something like this might be true. Some of you may have heard my lecture. It's so beautiful, something like it has to be true. But I said, I can equally guarantee you that exactly what I showed you today is going to be wrong. Because everything I've ever proposed at first has proved to be wrong. Okay? And so the same is true here, but something like it may prove to be right. So when you are in trouble, it means you're about to discover something. And we don't really know how to explain all of these facts today. And among the most troubling things is synaptotagmin forms a ring, right? Vesicles are curved. Well, you can see how that works. A ring at the base of the vesicle. But I just told you this complexin half-zippered forms a linear array. How do you work a line into a circle? Well, maybe the crystal is wrong. But there's a lot of genetic evidence, though not all of it, that suggests that it isn't, and very strong biochemical evidence. So we've been racking our brains recently. Fred and I and our friends and colleagues have been drawing up all kinds of models to try to bring this together. And this is kind of what we're thinking, okay? The genetics suggest, the surface force apparatus showed that a clamped state that correlates genetically can be formed that is half-zippered and stabilized with complexin and with snare complex alone. And the crystal structure revealed that it exists in a linear zigzag array. You can imagine how this zigzag array could be formed in such a fashion that it goes around and closes on itself. In fact, one of the criticisms before the zigzag array came several years before the complexin ring, and people and our colleagues and certainly ourselves were concerned what happens at the end of the array, because it can't be infinite when it's coming from a synaptic vesicle. Well, maybe there is no end because it closes on itself. And in fact, we have several different crystal forms of this array in which the geometries are different from one to the other. And so there is probably the flexibility for this to happen structurally. We know of no reason why this couldn't happen. Now, we also have the ring. And you remember I said that perhaps it does not accommodate snares at all, and the snares are just simply prevented from getting past the ring to fuse. Or perhaps its position, which we think is more likely. Okay? Now then, at the most, perhaps 10 snare complexes could be taken out by the ring. And remember I raised the question, what happens to the other 60? And what I'm suggesting here is if you have 60 V snares in the synaptic vesicle and many T snares in the plasma membrane, there's probably no reason why the plasma membrane could not be bent and that it could not then engage and form snare pins and indeed fuse laterally, sort of en passant, with the synaptic vesicle. And the energy required to do this actually is very modest. we think, as compared to the energy available in even 1 or 2 snare pins. So there should be no energetic problem for this to happen. So you can imagine then 2 entirely separate mechanisms coexisting in the same vesicle. If you do away with the accessory helix or complexin altogether, these snare pins are now free to fuse, and indeed they do, and they give rise to an excessive quantity of miniatures. But as a result of having complexin, this does not occur, and now the noise is eliminated, but the signal is allowed. Okay? Now, then we have the ring mechanism, and we might imagine that the complexin is also utilized in this ring, but maybe it's the central helix whose geometry is different, as I just showed you, between the half-zippered and the fully zippered state that may actually further energize or stabilize the half-zippered state, preventing zippering and therefore adding clamping as well as energy to this ring. And so what we think, to summarize, is, and I've made this very particular so you can picture it. I don't mean to say that it has to be like this, because it could actually be, by the way, that rather than being here, this zigzag array, circularized or something like it, could be here, and it may even interact with the ring, perhaps by the A domains. We don't know. So there may be 1 coordinated machine, 2 uncoordinated machines, but I'm struck by the fact that all of the biochemistry and all of the genetics can be explained by something along these lines. So this is the direction in which we're thinking. And a little bit more granularly, to summarize, what we suspect is that one way or another, numerous snare pins cooperate in a ring-like arrangement that is templated by synaptotagmin in one or another way and further energized by complexin. And what this does is to create a high-energy state in which the membranes are energized, and energized in such a way that they can be explosively released. And what do I mean by explosively released? Released in such a way that there's so much tension between these 2 membranes that it's a stressed, intense situation in which all of the forces are internally balanced as a result of the symmetry of the structure. And you can imagine, and if you picture it this way, that these membranes may be pulled together in this fashion, stressed, bent, that these snare complexes are constantly trying to motor towards the center, pulling these membranes together, held in this fashion. Pulling these membranes together this way will compress them. The snare complexes on the outside, perhaps these ones pushing in. So we don't know any of the details, but you can easily imagine a highly stressed, balanced state. And then when you break symmetry, and I would propose that symmetry is broken when the first molecule of synaptotagmin binds calcium and starts to leave the ring organization. Now the precise balancing of forces will no longer occur, and there will be a very rapid, potentially explosive release that will literally force the 2 membranes together, perhaps at a speed that would far exceed that which 1 snare by itself could accommodate. And so in essence, we could be taking the energy made available from a large number of snare complexes, some primarily, some secondarily, adding them all together cooperatively to make a large single mechanical unit that is perfectly balanced by symmetry, symmetry then being broken when the first synaptotagmin binds calcium. And let me point out a classic physiological observation. The Hill constant, the cooperativity, for the overall process of synaptic transmission is between 3 and 5, depending on who's measuring in the system. It's not a precise number, but it's in that range. Basically, 1 synaptotagmin molecule worth of calciums. So I would suggest that there's an amplification in which the first synaptotagmin out of the 15 or 20 that happens to bind its complement of calciums switches out of the ring, symmetry is broken, leading to a massive amplification, and a race has begun among perhaps as many as 40 or 50 snare complexes. And whoever crosses the finishing line first, which is why it's so very rapid, fuses the vesicle and releases many neurotransmitters. So a handful of calciums leads to many thousands of neurotransmitters in a single quanta. So that's the idea. And I will close with an illustration. You've probably seen this structure. And this structure, Notre Dame, is a perfect example, as is every building, which has a high degree of symmetry and a massive weight, of a principle known to architects even before there were architects. So here you have a structure that has an enormous weight. There are, in other words, enormous forces, in this case gravitational forces rather than snare complexes, forcing the structure down to collapse. But it does not collapse. Why does it not collapse? It does not collapse because all of its forces are internally balanced as a result of symmetry. Okay? What happens if you were to go in and remove the key buttress or some internal column, and you did it at just the right time and just the right way? And there are probably many of them that you could remove. What would happen, of course, is this building, which is inherently unstable, though perhaps stable it's certainly stable for centuries, we certainly hope millennia, but what would happen is it would immediately collapse, and you would release this potential energy as kinetic energy, the pressure would be relieved, you would create the stable state and symmetry as symmetry is broken. So I'm suggesting that something very much like this occurs, in which the bricks are essentially the lipids and the proteins that comprise a structure that through its own internal architecture self-generates force and self-generates pressure to create a physics that may not be entirely different to enable explosive release. So let me just thank the many colleagues who have contributed to this ongoing piece of work. It's a collaboration of several different laboratories. In my laboratory, the senior biochemist Shyam Krishnakumar, Jing Wang, who is the person who did the microscopy and discovered the rings, Oscar Bello, who did the mutagenesis based on the structural reconstruction and tested the disease mutations, Feng Li, who did the surface force apparatus work together with Fred and myself, and is now contributing to this project in other ways. A graduate student, Alexander Rebane, who is currently studying how complexin interacts with single molecules of the snare complex using optical tweezers in collaboration with Yongli Zhang, a professor in our department who is our optical tweezer man with whom the experiment that I described to you before and the last lecture was done. Karin Reinisch, an X-ray crystallographer, also in our department at Yale, with whom we collaborated on the half-zippered snare complex. Fred Poncet here in Paris, with whom we collaborate both at Yale and in Paris on this and related problems, and we hope to soon engage other collaborators here in modeling this process based on a detailed understanding of the forces and seeing if we can't come up with a soft matter physics inspired model that will enable quantitative predictions of the rate of fusion. Charles Sindelar, the structural biologist at Yale in our biophysics department, who I've already mentioned. I believe I mentioned Troy Littleton, with whom we did the fly physiology. David Baddeley is an assistant professor in our department at Yale, who is an optical engineer and biophysicist, with whom we are doing the super resolution imaging studies currently to see if we can visualize the rings in physiological contexts. And Kirill Velinsky is our collaborator, one of several at UCL in the neurology department on the providing cell physiological correlates as we move on. So thank you very much. There are others, obviously, who have contributed, but I won't mention them further because I haven't mentioned their work in progress. So thank you all very much.